It's a great pleasure for me to introduce you Silvio Salvi for the meeting today. He did his PhD in biotechnology at Bologna University in Italy, and then worked at the Research and Innovation Center in Trento on apple molecular genetics. Now he's back to Bologna, and I think by now a full professor there, right?
Yep.
Right.
Yep.
Working on the genetic of agronomic traits of different crop plants, among others tomatoes and grasses, and also cereals like maize, durum, wheat, and barley. And he manages different mutant collections, one of them of the bioenergy grass giant reed, and the other one, which he is here for today, the Tilmore collection, which is a collection of mutagenized barley in the cultivar Morex. And he's going to talk about that today, and I'm especially excited because he's going to focus on roots. So the stage is yours, please go ahead.
Thank you. Thank you, Gwendolyn. I like also to thank Timothy George and Robbie Ball for first asking me to give this presentation within this nice network of scientists. And so, yes, I'm going to tell you a little bit of our advances that we are doing in the trying to understand the root biology, especially root development in barley, using this mutagenized population. Okay, so in perhaps I should know this one. Okay. So in barley, we recognize several different root types. Starting from developing seed, we see the primary root, which is basically the first little root coming out from the embryo. And then very soon after this first developmental phase, we have seminal roots from primordia already present in the scutellum zone around the embryo. And all these roots are basically pretty much the same, with the same biology, the same physiology, the same anatomical architecture. That's why also this root system is called homorrhizic, so made of similar roots. And then later in the development, from the shoot, from the first nodes close to the seed, we have the development of the so-called crown or brace or nodal roots, depending on the species. And which are eventually at the end they will constitute most of the root apparatus during the adult phase. The third type of roots are the so-called lateral roots, and lateral roots are defined as the roots developing from other roots. And so we will eventually have lateral roots from the primary, from the seminal roots, and also from the crown roots. Of course, root traits are of major interest in crops. And not only for very basic biological reasons, because they constitute half of the plant biomass sometimes, because they provide nutrient and they provide water to the rest of the plant, but also for some specific reasons that might also be overlooked by scientists and by agronomists. I tried here to make a kind of very general school text list of traits that are interesting and especially interesting for geneticists and for physiologists, for root physiologists. So we start from the root shoot biomass rate, which is more or less important, especially it could be also very, very much important for those species where the root is actually the harvested crop, the yield like sugar. Then we add the function of anchorage to soil and root lodging, and then the general root spatial architecture, which is defined by trait components like the number of axial roots, the number of lateral roots, the root growth angle, the root distribution, the soil profile, whether it's in the upper phase of the soil or is more down deep in the soil, the growth speed, the circummutation, and so on and so forth. Then we have anatomical traits, which are pretty much really exploited at the moment and underexplored at the moment. And we, besides the very, very famous Irencema type of root aerial spaces in the root, we also have the number of size of xylem vessels, which are probably under genetic control, and etc. Then we have root hairs, then we have root exudates, and which are very much important for soil carbon allocation, but also for the interaction with the rhizospheric microbiome. And then we have a number of physiological traits like the capacity, the ability of the roots to uptake nutrients or water, and etc. Of course, I'm going more in agronomic perspective, and because this is a pretty much an interesting trait for our lab, for our group. Roots are particularly important for drought tolerance, which is an important aspect of climate change that we are all experiencing. And as you can see in this very recent, very knowledgeable review about the importance of the roots and the mechanism of the plants used to address drought, you see that a very large room, a very large space is dedicated to root traits like root architecture, as we have already listed, like the rhizosphere traits affecting soil matrix potential, rhizodeposition, root hairs, and so on and so forth, and then root hydraulics, which are pretty much affected by the anatomical structure of the root, besides other aspects. So what we know, going more closer and closer to barley, what we know about the root genetics in barley. So we know that barley is particularly interesting because there is a very large, there are several different large collections of mutations, and but as we have already discussed into this community, while many of these mutations are important for are present and described for the shoot part of the plant, very few information, very little mutants actually are available for roots. Historically speaking, at least in the barley genetic newsletter, besides the many hundred mutations for the shoot part of the plant, we only had one mutant described for the barley root architecture, which was called FER1, so fewer root 1. And by discussing with Uda a few years ago already, when I asked for this mutation, for the seed of the mutation, I was told that this actually the only mutant described in this community, in this network, was actually lost. So at least until a few years ago, the barley genetic community was without any barley mutants, at least when we address the root architecture, the general root architecture. Indeed, there is a nice few collections of root hair mutants, mainly put together by a few colleagues across Europe, especially Professor Foster in UK, and Professor Zareiko from Poland. So we ended because of the general interest of the lab toward the genetic mechanism to inform us and to provide us solution for drought tolerance, we decided to exploit our barley tilling population in this direction. We developed our tilling population already many years ago, and for the specific purposes of a tilling population, so for reverse genetics, we developed the population using the Morex cultivar, because already at that time it was pretty clear that Morex would have been chosen as the reference cultivar for the first exercise of genome sequencing. We mutagenized the Morex seeds of Morex using sodium azide, and we developed a population of about 5,000 lines, which are now reduced to 3,600 after a few generations of seed reproduction. We killed several genes for in a real idea of specific, reverse genetics perspective, and we found several alleles, different alleles, and a large number of missense or knockout mutations. We were able to estimate, based on this tilling effort, the mutation frequency in the population, which was estimated as one mutation in approximately 500 kb, so approximately 2.1 mutation per megabase. Considering a few computation, making a few mathematical computation here, we estimate that we have 10,000 mutations per line, about 6 in exons. I jump now in 2024, so almost 20 years after, 15 years after, and because we have been sequencing several of these mutant lines, now we have a better estimation of the mutation frequency, and the mutation frequency based on 25 whole genome shotgun sequencing, and using Morix as a reference and annotated line, we estimated that one mutation out of 250 kb, so about 100 mutation in exon per line. We were able to estimate the presence of about 3 knockouts stop codon, so gene completely knockout per line. This at least, I should say that this is at least 3 knockouts per line. During those years, we also realized that besides exploiting the population, as I said, as reverse genetics, there was so much phenotypic variation visible when we were growing the plants in the field that we decided that probably it could be a good idea also to screen the population in a forward genetic fashion, so starting from the phenotype. So this is what we did for different phenotypes for the shoot, but today we are talking about roots. So we screened the population with several different tools, starting from the very simple paper roll to plastic sandwich, semi-hydroponic system, and up to soil field rhizotrons. You can see here a magnification of the paper roll system that we used. As you can see, of course, everybody knows this very simple approach, which this notwithstanding allowed us to really identify the major mutations that they were really informative later on in the story, as we were able to clone the genes behind those phenotypes, those simple phenotypes that you can see using this phenotyping approach. And here, for just an example of an open paper roll with 12 plants, and where you can see already a very clear short root phenotype, pretty much specific for the root and not affecting the shoot. So these were the phenotypes that we always been looking after, or going after, because of they were showing some specificity for the root. The second system that we used, and we are really using now a lot as our major system to screen this population and other populations, is this acrylic plastic plate sort of sandwich, where we use the plastic plate as a support, and then we put 2 filter paper sheets on it, and in between you put the seeds. This can be done horizontally, as you can see in this picture, but also, vertically. But also we can turn it 90 degrees and using horizontally in order to screen more seeds, but for a fewer days. Everything is moistened in via ionized water and then grown for a couple of weeks, no more than 2 weeks in standard condition. So you see possibly a picture of what this end system is. This was very, very, very informative and very, very good. In order to increase the throughput and perhaps losing a little bit of new information, but throughput and specifically numbers in genetics are important, we miniaturized our approach and we went down to the compact disc CD cases. I have to translate this and say CD, that means compact disc, because many of the younger generation don't even know what this stuff is, but it is much more meaningful for people of my age. And so these are basically a 10 by 10 centimeter square plastic box where the compact discs were kept, and we put some filter paper in it, and you can grow 3 or 4 seeds for each of your line for 3 or 4 days, no more than that. But this is already enough to see many interesting phenotypes and really to screen a very large number of plants in a very short time. And the soil field rhizotron, there is not much to say, except that we really standardized this phenotypic screening in our lab, in our team. So it's pretty much standard rhizotrons with one glass wall and the second black aspect wall on the other side, and some shims to make some room in it, and binder clips to keep everything together. Here you can see the settings. And in order to collect the images at the end, we use A3 scanner, flat scanners. We have 3 of them, and also to provide some throughput in this work, which is, on the other hand, completely manual. So by using this approach, now I'll tell you some successful stories quite briefly, actually, because I like also to present some newer data. And starting from really the paper roll system, by observing some original kind of strange behavior of those mutations, we were able to select a couple of promising and possibly hypergravitropic mutants. Here you see already the mutants after the first selection in a paper roll, regrown in using a flat system, in this case with blue paper, and you can see the phenotype. So this is the mutant line, homozygous, and this is the wild type, in this case Morex wild type. And the mutant line was TM2835, and here you see already with the name of the locus that we decided to give, so enhanced gravitropism 2. Already by the number, you can understand that we have also a number 1, and actually now we have very likely number 3. Here you see the phenotype using magnetic resonance with the help of colleagues from the ULIC station, or the research station. And you can see that the gravitropism, the hypergravitropism phenotype that we observed was not a passive phenotype, but it was actually an active and very, very quick regrowth of the root apex in a new direction. Here you see the very famous rotation test that we can use to test this hypothesis. Here are the wild type 1x here, and 0 time, well, before, just a few days after germination and before the rotation. So this is our mutant, which is growing perfectly down, gravitropic, in a gravitropic way. And then after the rotation, you see that the AGT2, TM2835 is much, much more quicker in getting again to the perfectly gravitropic direction in comparison with Morex. So the gene was cloned using a pretty much standard bulk segregant analysis using an F2 population developed by crossing our line with a different line in order to leverage the SNP polymorphism between the 2 background lines, so Morex and Barke, and using a high-density SNP array in order to genotype the 2 bulks. As you can see, we got a very nice single peak on chromosome 5, and mutate after sequencing, whole genome shotgunning sequencing of the mutation, finding a gene mutated in that region, a single gene mutated in that region. This gene was encoding for a sterol alpha motif protein, and we were actually able, with the help of Gwendolyn, this was led by actually our lab in collaboration with the Franco Koldinger lab with Gwendolyn Christian, who actually introduced my seminar today. And as you can see, we only had 1 line and 1 allele from our Tilmore population. So in order to prove that we are after the right mutation, we generated a second allele using CRISPR-Cas. And so here we have the mutation from our Tilmore, and the second allele was generated as a deletion in the first exon. And by growing, again, with the same type of phenotyping protocol, the CRISPR-Cas generated line, the phenotype was the same. So that proved beyond any doubt that we are after the right gene for the mutation. Very nicely, we tested whether the phenotype was conserved in all the types of roots that we have in barley, as I said at the beginning, and definitely yes. The hypergravitropism phenotypes is conserved across seminar roots, crown roots, and also lateral roots coming out from the axial roots, as you can see here. Quite interestingly, we didn't see basically any obvious effect on shoot traits like leaf angle or seed spike phenotypes, etc. In order to understand what's happening after the mutation of this gene, which is a protein basically involving protein-protein interaction, so not really by itself, not really easy to be connected with the root phenotype. An RNA-seq analysis was carried out, and by dissecting roots in different positions, the root cap and the meristem region and in the elongation zones. And to make a long story short, we found 76 differential expressed genes between EGT2 and the wild type, and those 67 genes, 7 expansion genes, which are already known to be involved in the cell expansion, and especially in the elongation zone, which is a prerequisite to have an gravitropism effect were identified, and then also peroxidase identified. And this is also very well known that we have pH changes in the cell wall during the process of root bending and root growth. And indeed, we also identified some link with the auxin physiology of the root in this gene, exocyst complex component 7, which is linked to PIN transportation protein. It's actually was nice to see that our gene is one of the close homologue of the WIP gene, which was cloned a few years ago in peach. The WIP gene is actually expressed, so you see the phenotype in peach, in the peach tree, in the shoot part. And the mutation in EGT2 homologue in this WIP gene is causing this bending phenotype, which is not a floppy bending, but is actually a strong rigid type of shoot that you have in the phenotype. And this is actually what we do observe also in our root mutants. Our root, the roots of the mutants are not floppy, but they are, stiff root, really going in a hypergravitropic way. As I said, using the original population, we found additional hypergravitropic mutants. And this is another one that we found, and that was published more recently, in this case, in a collaboration with our group and the group of Nottingham University, Malcolm Bennett's lab. And as you can see, the phenotype is pretty much the same of EGT2. So this is a different line by X-ray microcomputer tomography, and here with virutrons. You can see that also in this case, the hypergravitropism is basically the same as before, so expressed in both in the seminar roots, which are the green lines, and also in the lateral roots, which are the red lines. Using the same approach, we mapped the mutation on chromosome 6, and turned out to be the causative gene was identified, and it's a double domain protein, an F-box double domain protein, and again with a not clear, obvious association with the root phenotype, which is a good news because then in the two cases we are able to find new genes affecting root architecture, which were completely unknown from the vast amount of studies that they were done before in Arabidopsis, mainly in Arabidopsis. And in this case, by screening the population, the tumor population, we were able to find a third line, which is pretty much a similar phenotype. We sequenced third line, we were able to find a mutation in the same gene. We also carried out complementation test. And so in this case, the final proof that we were after the right gene was done because we found two different mutations in the same gene in two different lines. So what we learned in this work. So basically, I believe that what we carried, what we understood, and it's that we opened the door in a mechanism of gravitropism, which was actually not very well known before, as I said, based on studies mainly carried out in Arabidopsis and dicotyledon species. So we all know very well that the gravitropy is perceived by the root tips in the columella cells in this region, and by the presence of the transform cytocystis chloroplast, which are perceiving the root gravitropism force, and they are moving into the cell. This sends in some unknown way a message to auxin protein, transporting protein, and in a way that a large flow of auxin is at the bottom of the roots in this case, and a much lower flow of auxin is actually resulting at the top of the root. This eventually end up in decreasing cell elongation in the elongation zone in this area, and increasing cell elongation in that area, producing the turning of the root tip toward the gravitropic force arrow. So basically, and then root will continue to grow in some sort of very well-defined gravitropic set point angles. So this is well known for people working on root. Roots don't really grow randomly, but they actually grow for at least for some time to a very well-defined gravitropic set point angle. So the results of the final angle that the root is using to grow at least for a certain time is defined as the gravitropic set point angle, and must be the result of two antagonist forces. One, the gravitropic force, which is the one that was known before, and a second, anti-gravitropic response, pulling the root somehow in the other direction, so upward, in order to keep the root growing in a defined gravitropic set point angle. And indeed, the discovery of EGT1 and EGT2, which when they are mutated, are not providing the anti-gravitropic offset force, really opened the door to understand which are these forces pulling the roots and keeping the root growing to a defined root angle. Without this type of cellular functions, so again, the anti-gravitropic offset mechanism, all roots would grow perfectly vertical down. And based on the RNA-seq and some other additional studies that have been carried out also in the group of Franco Colde and later on, it was pretty clear that more or less the EGT2 and EGT1 and EGT2 are actually acting in these pathways by regulating peroxidase and cell wall loosening enzymes like the aspansins, as I said before. And perhaps, at least for EGT2, we have a connection with pin proteins reposition that we were not able to see, at least so far, with the EGT1. All of this will actually cause in this anti-gravitropic response, which was pretty much unknown before. I mean, these major players in this anti-gravitropic response were completely unknown before. And we carried out, and because of the similarity of barley and wheat, one interesting hypothesis that we wanted to test is whether these mechanisms were conserved in wheat. So we took advantage of the tilling population available in John Innes, and with the help of the group of Cristobal Huawei, and we identified the triple mutants in cadenza, but also the double mutants in the tetraploid wheat chronos, and the phenotypes was pretty much conserved. As you can see here, the triple mutants, homozygous mutant above, and the wild type below. So very nice result for EGT1 here and EGT2 here. So the two mechanisms are conserved, and so this is a good news for exploiting these mutations or these genes for plant breeding purposes. I'll tell you now a little bit more. So concerning EGT1 specifically, we tested the possibility that to see this an effect of the genes already as a QTL, so across native genetic variation. We did that by screening a large and representative collection of lines in collaboration with the WellBe project, and in collaboration especially with Luigi Cattivelli, one of the partners of the WellBe project. So we tested the full population for the same phenotypes that we have been discussing so far. And that was really, really informative because we found so many very large variation, in this case, native variation from different cultivars, and providing, you see, phenotypes as extreme as the mutations that we identified in the timber population. Both for just showing you two examples for root growth angle and for number of seminar roots. But interesting enough, so taking advantage of the availability of the exon sequence, we tested the allelic variation for EGT1, and we found that by putting together the allelic variation of EGT1 and the root angle phenotype that we collected in the same population, there was a clear effect of EGT1 as a QTL on in the WellBe population. Okay, move on. We as I said at the beginning, we wanted to get more high throughput, so we decided to switch on to rescreen the population, which is now a little bit more than 2,000 lines, using the CDKs-based protocol, for more or less the same phenotype, or let's say for all the possible phenotypes that you can screen using this approach. So far we screened, as I said, a little bit than 1,000 lines, and using this protocol, which is pretty much a very quick protocol. You do everything in basically 1 week. I don't think I need to waste a lot of time here, but as you can see, we ended up in identifying a new hypergravitropic mutant. Here you see the phenotype that we first discovered in the CDKs compared with Morex. Then we did a rotation test. Morex, rotate slowly, while this new mutation rotates much quicker. Then we moved to the plastic plate, and then again the phenotype was fully confirmed. We also tested the rhizotrons, Morex and the mutant after 7 days, and Morex after mutant, and the mutant after 21 days from germination. You can see that again. Now, well, of course, here I switch the two. Sorry, I switch well, it's pretty clear that Morex is on the right-hand side and the mutant is this one. I apologize. Anyhow, you can see that the phenotype is fully expressed in the seminar and also later on in the crown, which you don't see them here, but also in the lateral roots. Quite incredibly, so let me say so. By screening our tilling population, we ended up in 3 different mutations affecting gravitropism. You see Morex, you see EGT1, EGT2, and the new gravitropic line. We have already done all the tests. This is under publication. Let me for once not. We are still basically understanding, testing making the last experiments in order to know which is really the gene, but we know already it's a different gene from the first 2. So incredibly, 3 different hypergravitropic mutations, independent from one from the other. In the last couple of years, a couple of very, very talented 3 students from our lab decided also, against my wish, to screen the population also for root hairs. But this was really informative as well. So I'd like to show you some of the latest results. So based on the old screen using Pepperol and the new screen of the 1,000 lines using the CDKs, we identified a number of preliminary and possible candidate mutations for a mutant for root hairs, which were then confirmed using the rhizotron, the soil field rhizotron protocol. In this case, we use a very simple handheld microscope, and you can see here the type of pictures that you can get from this very simple and cheap approach, so very informative. We ended up in having finding 13 mutations for root hairs. Here you can see here a quick overview of the mutation. We are trying to publish this list right now. So we have mutations, root hairs mutation of all types, mutation that interestingly don't show root hairs in the seminar roots, but then they show root hairs in a little bit in the lateral roots and fully in the crown roots. So here, a very interesting and completely new phenotype never observed before in other species. And then we have, small kind of abnormal root hairs, mutant, completely naked lines, so without root hairs, and so on and so forth. And we have 13 of them. Here you see an overview of the pictures. Here on the left-hand side, you see the phenotype, the standard phenotype of the Morex, which was always tested as tested. And we had just completed, or we are completing these days, the photographic description of the mutants, and by adding also scan electron microscopy image in collaboration with, again, with the lab of Franco Goldinger. And so we are adding here a best representation of the mutants. So here you see an example of short root hairs, and here another example of root hairless. So we should be able to provide this to the community very soon. And let me say that all the mutants, including the completely root hairless, are completely viable, and in standard condition of growth, like good humidity and good fertilization, they basically do not show any phenotype compared to Morex. We also addressed the very common mutations that we find, which is the most common type of mutations, which is the short root. Short root doesn't mean much. You have a short root anytime you have a weaker plant, so compared to Morex. So we fully understand that going after short root mutations might end up in going after something that is not so informative. But nevertheless, we put a collection of short root mutations, and we are actually now trying to clone some of the genes behind that. I can tell you that these two, just to give you an example, these two short mutations ended up to be allelic of one interesting new gene, which is an oxytransportation genes, providing very nice anatomical phenotypes, informative about the anatomical development of the root, of the barley root. I'm not giving you information today about this, but I'll give you some information about this other mutation, which is from the line TM390. Here you see the phenotype. This is a wild homozygous mutation. This is the F1, and this is the wild type Morex. And here a magnification of the mutant. So the mutant, at least in the very first phases, is very much root specific. The root basically do not develop while the shoot is perfectly normal. Again, we are in the process of mapping and cloning this gene. We run the full mutant, the full bulk segment analysis, and here you see the result. The gene definitely map on chromosome 2 in the pericentromeric region, so the signal that you can get by putting the physical distance on the x-axis is basically of no information, because most of the chromosome 2 region is actually pericentromeric, so there is no recombination, but we sequenced the line and we found a very promising causative mutation in this region. So which is a very interesting gene affecting root development. So also very nice information that you can get by apparently non-informative mutants like this one. What is the current root mutant collection situation? So as I said, we found 4 hypergravitopic lines corresponding to 3 different loci. Quite amazing, in my opinion, that I couldn't really believe that. And then we found 23 short mutant lines, about 4 disturbed root mutants, probably affecting the wavy in the in the way that the helicoidal growth of the roots is doing. And then, as I said, 13 rooter or rooterless mutants. Let me finish in making a list of the additional traits that we have under target for the future projects. We have been funded for carrying on additional root genetic analysis in barley by our ministry. We are starting during literally these days the new projects, and so we would like to address novel root traits which have been hardly addressed so far by the scientific community. We would like to address root circummutation, so as I said, the helicoidal growth of the roots in the soil. This is a very important trait affecting how efficiently the root is exploring the soil. And this is basically not have been addressed so far, except for a couple of genes that have been found in rice and Arabidopsis by the group of field benefit. And so this phenotype is probably involved at tens or perhaps hundreds of genes, completely unknown at the moment. We would like also to address the dynamic of the growth set point angle. It's very well known that the growth set point angle is actually changing during time and space, and for instance, why the roots is getting older. And the function and the genes affecting this very interesting phenotype, very important also from the agronomic standpoint, are completely unknown. Of course, root traits are affected by the interaction with the rhizobiome or by the interaction of other plant roots. Also, basically, no genes known to be involving other plant roots interaction are known, except for the ones involving the recognition of the parasitic plants. Basically, nothing is known about the mechanism of root senescence, and which are interesting for annual plant, but probably even more interesting for perennial species. And of course, this very famously right now during this time, the genetics and the physiology of the exudation affecting the total carbon sequestration and emission. With this, I like to finish, and I really like to thank many of the collaborations. As I said during my presentation, and the cloning of several of these genes were done in collaboration with both the group of Franco Coldigan from Bologna University and the group of Malcolm Bennett at the University of Nottingham. And many of the additional experiments were done in collaboration with John Innes, with IPK, with CRI-FEM Fiorenzuola and the NIAB at Cambridge, UK. And the major players in our group for the results that I presented today are Giuseppe Sangiorgi, Alberto Tassinari, and Serena Rosignoli, and Christopher Enstan. And let me say that I forgot to add Silvio Perbattista, a very recent postdoc who was able to participate into the root hairs study. Thank you very much.
Thank you, Silvio. That was extremely interesting.
Thank you.
I'm going to read out some questions from the chat. So please add more questions or maybe raise your hand. I don't actually know if I would see that. Maybe add it to the chat.
Shall I keep this presentation? Shall I show? What should I do?
Up to you. I can see my chat. So one question from Niels is if the EGT1 and EGT2 mutants are additive, and if you check the double mutants.
Yeah. Yes, we checked the double mutants, and the result is you cannot be more hypergravitropic than the perfect hypergravitropic. So, once they are so perfectly vertical, they are perfectly vertical. That's it. Yeah, so this is true. Yes, we did it. We did it because we wanted to, from the beginning, we didn't really believe that we had two genes. As I said, now we have even three, but we didn't even believe that we had two different genes. So the complementation test was our first experiment, yes.
And Neil's second question was if what are the natural diversity haplotype differences in EGT1 and EGT2 correlated with the root phenotypes?
Yes, indeed. So now we have also completed a full genome-wide association analysis for the traits, which were not completed at the time of EGT1 publication. But I can tell you again, I can confirm that the EGT1 is visible. So you can map EGT1 as a GWAS skip peak, but not EGT2, at least in our in the WellBe collection. That doesn't mean that in other germplasm collection you might have allelic variation in EGT2, and you might be able to have it some effect, so some QTLs. But at the moment, at least in the WellBe population, there is no allelic variation. This was also proved by analyzing the sequence of EGT2 and EGT1. While we had several different substitutions, variations, so nucleotide substitution in EGT1, we had very little in EGT2 and basically not affecting the protein structure.
Okay, Robbie's question, one of Robbie's question is if you grew the EGT2 mutants under different soil stresses and if there were performance-related phenotypes.
Yeah, this is, of course, this is an important question. So far, we did the experiment several times, but we are in the process of completing the backcross of the two lines in order to clean the genetic background from additional mutations. So until this process is not complete, I believe that any conclusions about the pleiotropic effect of having this mutation, positive or negative effect of this mutation on plant performance are not really reliable, because as I said, we have tens, well, hundreds of mutations in the lines, and perhaps several different additional knockouts. So I cannot really answer. So what we are currently doing is to test whether there is a difference in root depth. You expect that the two hypergravitropic mutants, if all the rest of the physiology is the same, they should reach deepest soil areas, no? But this still has to be proved. This is more difficult than you can think of.
And the next question in the WellBe collection, was there a correlation between the haplotype, the phenotype, and the geography?
We take a look at that. We took a look at that. Unfortunately, the most extreme alleles were basically spread all over the geographical locations. So there is no strong geographic effect for EGT1. However, I can tell you that as I briefly show in my presentation, we found extreme cultivars showing extreme gravitropic behavior, and those were particularly, although the numbers are very low, anecdotally, I can say that they come from very dry environments. But the numbers of the extreme phenotypes are actually very, very low. So making any extrapolation is probably not correct at the moment.
Okay, there's one comment in the chat that asks for collaborators. Maybe you want to address that person later. And one question. First, thank you for the talk. And have you tested those gravitropic mutants for heterotropism as well?
No, we haven't. I know there are protocols available, and that is something interesting that we might want to address, not only for water, but also for nutrition. We would like to see how much the gravitropic response is disturbed by the availability or not of nitrogen. This is actually in the focus of our next project funded by our ministry.
Yeah, and a question that was relating to a question before. What is the relevance of these gravitropic mutants for drought response? But I guess you've already answered that.
No, but it is, I mean, I can briefly repeat it. I mean, it's based on ideal situation. If only this gene is affected and this trait is affected, definitely we expect that the mutant should reach deeper layer in the soil. And In most of the, I mean, not in most, in many environments, that means reaching more water. At least in the environment of, let's say, temperate Europe. Then perhaps in other environment with extreme dry conditions, the opposite. Most of the water is in the upper phase because you have very little rain or you have irrigation.
And the last question for now, which is more the material and methods part of the whole thing. Which software did you use for measuring the root traits from the images?
Which software you said?
Yeah.
Oh, we have the full list of software.
Okay.
Full list of softwares that, yeah, I should say that the softwares in this case, let me say something maybe trivial, but working with mutants with such as clear phenotypes, I mean, the collection of the numerical phenotypes is actually a matter of being clear, and but the phenotypes that we dealt with in these studies were really very clear. And but indeed, having good softwares helping you to collect phenotypes are good, especially when you work with more quantitative phenotypes. So in GWAS perspective, I do agree that it's very important to use softwares. I don't know who asked this question, but please address me directly on the email, and I will make you the full list of softwares that I can advise.
Great. And maybe the last question. Why were EGT1 and EGT2 not found in Arabidopsis already before?
Yes. This is kind of revenge, no, of our community. Anyhow, now they've been found. Now there are indications that after we published together with you, Gwendolyn and Riccardo and the other guys for the EGT1, now they're finding the effect also in Arabidopsis, and very nicely. But the reason, in my opinion, is that it's because of the root architecture of the two species. In Arabidopsis, you have this taproot type of system, so with the root getting down, and you are left only with the lateral roots in order to be able to see a root bending phenotype, no, and a gravitropic phenotype. While in barley, in wheat, or in maize, not only you can exploit the lateral roots to see a gravitropic defect, but you can also see directly already with the seminar roots. So I believe this is the very reason. So of course the mechanism must be in play also in Arabidopsis and in the dicots, but simply it was much more difficult to see that and to spot it by screening mutations or mutants or natural variation. So that is also good, that we do need not only one or two models, but we probably need many models to reach our complete understanding of root biology.
Thank you very much. So there are no questions anymore in the chat. If there are no more questions, Robbie, do you want to announce the next seminar in 2 weeks, because you've just put the message to the chat?
Sure, it's in the chat if you want to actually read it, but it's basically mutagenesis again, which is great, for barley gene discovery, genomic selection, and genetic improvements of Chinese barley varieties. And it'll be a bit, I can't remember if the time is different. I think the time is different, and it'll be by Ping Yang and Gan Qi Zhao. And I apologize for my Chinese. Yeah, that's it. 2 weeks time. Feel free, and I hope you can all join us there. All I wanted to say just to finish was, Silvio, that's fantastic, great stuff. Thanks so much for your presentation, and hope you can join us maybe in 2 weeks' time.
I will.
I'm sure you will. Okay. Lovely to see everybody. Take care.
Thank you.
Thank you, Gwen.
everybody. Bye-bye. Take care.
Bye-bye.